Und Beñat Agirre Mindegia, herzlich willkommen, der euch jetzt den letzten Final Slam präsentieren wird, ist ein solcher. Er spricht zwar wunderbar Deutsch, aber dieser Slam wird trotzdem auf Englisch sein. Und er promoviert in der Medizin, in der Biomedizin, und sein Talk is all about how to talk to cancer cells. Enjoy.
Okay, so hello everybody. My name is Beñat Agirre Mindegia, and I'm a PhD student at Uniklinik RWTH Aachen. And as you might notice from my accent or my name, maybe I don't sound very German. So I'm going to talk about that. Well, not about exactly how to talk German, but I'm going to talk about something also similar, how to be in an environment where you don't speak the same language. So no, I'm not going to teach you how to learn German. I'm going to teach you how to talk to cancer cells, which is as hard. So, but before, let's start from the beginning. So how did I end up? What's my journey from far away until Aachen? So everything started here, in a northern region of Spain called the Basque Country, more specifically in a very tiny village in the Pyrenees called El Zaburu. Here, as you can see, there's not much, but that's not true. There's a lot of nature, and that's what made everything start. So I was very keen to understanding how my surroundings worked, and that's how I started to decide, okay, I want to study some science, some biology. But for this, I had to move to a little bit of a bigger place. And for this, for my bachelor's, I moved a bit more south to the coast of Catalonia, to a city called Tarragona. This city, it's a very historical Roman city, and here I had to make a big decision that most of you will have to make also in the next 1 or 2 years. What to study, right? It's not an easy choice, and I couldn't decide between 2 bachelors, biochemistry or biotechnology, so I just decided to make both at the same time. Maybe not the best idea, but I somehow survived. There was something else I wanted to also do apart from studying, and that was living abroad, learning new languages and learning new cultures. And for this, there's something called Erasmus, which I very strongly advise you to do if you can. And this brought me to the elegant city of Vienna, the capital of Austria. Here, I got into contact with German, or something that they say is German. And here, I was like, okay, now I want to also learn German, which is, as you can see, I don't get the best decisions for myself. But anyways, for this, I had to look for a nice place in Germany, and I looked for nice livable towns around here, and I found Aachen, and I really liked it. So I was looking more into, okay, what can I do there? So I was looking for research institutions, and I bumped into the RWTH, which is a quite strong international university, and more specifically, looking for bio-research, I bumped into the Uniklinik, more specifically, the group of Professor Antonin. Here, I was very interested on their research projects, and I decided to enroll for my bachelor's internship. I did it here, I got the place, I really liked it so much that I decided to do a master's with them too, and now a PhD with them again. So that's how I got here, but what I'm going to tell you now is why I got here, what's my project about, and let me tell you the story of how my project started. So I work with myeloproliferative neoplasms. Horrible name we will call MPNs from now on. And this disease is a group of cancer cells, of blood cancers, that might sound scary, but patients that undergo treatment can live with it for long and healthy lives. But there's a problem. Some patients, sometimes, develop more severe cases of blood cancer, and then they have fatal outcomes. So we were asking why this happens, because it's not known yet. So we had to make the big question. Every research project starts with a question. How does this happen? So we were like, okay, what's wrong with these cancer cells? So we thought maybe they were dividing wrong, and then these errors were accumulating, and this was causing this change and these new cancers to develop. So I know your teachers told you this was a school-free day, so I'm going to be quick in this, but I want us to be on the same page. So, cell division, also known as mitosis, you will also already know, or you will know after this slide, hopefully. For a cell to divide, we need to first take the DNA, make a copy, pack it into chromosomes, put them in the middle, and then spread them into 2 new cells, and then we have 2 nice new cells that are exactly the same as the mother cell. So errors in this separation is already understood as a very important part of cancer disease. So now, it was the moment to ask the big question. Let's ask the cells, how are they dividing? So as you can might guess, I didn't get a very clear answer from it. Sadly, cells and I, we don't speak the same language. So then we got into thinking again, how can I look for the answer myself instead of asking? And this, we thought, there are tools in the lab to find how cells are moving around. So I could go to the microscope, and I went to the microscope, but this is an optic microscope, which only uses normal light. And when I zoom in on the cells, I can see the cells from the patients, but I don't see any chromosomes. So how can we see this DNA? How can we make these chromosomes visible? And then we thought, okay, there's another kind of microscopes, these fluorescence microscopes, that allow us to do so. For this, we have to give some color to the DNA, as the DNA is not visible for the naked eye. This is done by a molecule that binds to DNA, and this molecule is very special, because when you point it with a very specific laser, this molecule will shine. And we can take a picture of these shining things, and this will be exactly where the DNA is. So we go to the microscope, we put the cells into a cage, they don't like it so much, and then we tune all these factors, which laser to use, how strong this laser should be. And after a bit, we get a nice picture where we can actually see the DNA of the cells and even the chromosomes. So now the only thing that's left is to take videos of these cells dividing and then look at them very into detail if this division is done properly. So here we have a nice video of the cells dividing. We have the chromosomes in the middle, then they separate, and we have two new cells with new nuclei. So this will start again so you can see it better. Chromosomes are there, then they are lined in the middle, separated into two, and then we have two new cells. Perfect, we can see it. Now let's have a look at this other video, for example. Same process, right? Looks exactly the same. In the middle, we pull them away, and then we have two new cells. Perfect. But if we actually stop the video and make a closer picture, and these are 4 different cells that I took as example, and we zoom in, you can see that this is not done so well. You can see in the white arrows, in the first and fourth picture, there are some pieces of chromosomes, or like these red dots, that are not in any of these sides. If chromosomes go to the wrong side, one cell will have one more chromosome, the other cell will have one less. This has fatal or very severe outcomes. Also in these 2 middle pictures, we have sticky chromosomes that when separated, they don't want to go away, so they just have these little chewing gum threads that break at some point because of the pressure, and this can also separate genes into different directions. So, okay, this is all cool, and we could see that patient cells actually were dividing worse than healthy control patients. And then let's translate this into what actually happens in the patient's blood, because this is a blood cancer, right? So here, this is a very simplistic cartoon where we have happy cells that are actually sick from MPN, but with treatment, they are quite fine. But then they start, with time, accumulating these mistakes. These mistakes make the cells not be so happy and have more probabilities to evolve into worse and more severe cases of blood cancer. And at the end, we get this outcome, or that's what we thought. So this is a very preliminary thing, so there's still a lot to do in the lab, and that's why we are always there. But in the power nap between experiments, I dream of actually finding the solution for it, and why not, how to stop this from happening so that patients can live healthy, long lives without having to worry about other cancers appearing. And why not, dream big or go home, right? Winning the Nobel Prize for Medicine. Okay, so this is the story. This is my story on how I learned to talk to cancer cells. This is my journey also, and this could be yours too. But it doesn't have to be. You could choose your own way. We are just here to say that science can be very much fun, and if you have the opportunity and the passion for it, it can be stressful but very satisfying, and at the end, it can bring into making a difference for society, which is always the best goal. So, nach fast 2,5 Jahren habe ich geschafft, mit Krebszellen zu sprechen. Und auf dem Weg habe ich ein bisschen Deutsch auch gelernt. Also, obwohl am Anfang so schwierig aussieht, man kann auch es schaffen. Sogar wenn auf ein sehr kleines Dorf in den Pyrenäen man kommt. Vielen Dank für Ihre Aufmerksamkeit und wir sehen hoffentlich bald uns im Labor.